Buenas tardes a todas, a todos. De parte de la Sociedad Argentina de Inmunología, de la Comisión Directiva, les doy las gracias por estar hoy aquí, junto a nosotros, en este último jueves de inmunometabolismo. Quisiera dar gracias particularmente a Mercedes Borge, a Luciana Balboa y a Cintia Stempin, quienes organizaron esta serie de webinars sobre este tema tan fascinante para todos nuestra comunidad. En el día de hoy nos acompaña el doctor Edward Pearce, que va a ser introducido por Luciana Balboa. Y bueno, muchísimas gracias a todos por estar hoy aquí. Espero que se sigan sumando a las actividades que organiza la comisión directiva, y los dejo ahora en manos de los organizadores del webinar. Muchas gracias.
Okay, thanks, Mariana, for this kind introduction. I will then start by presenting our guest speaker for today, which is Professor Dr. Edward Pearce. Dr. Pearce pursued his PhD at the Division of Parasitology at the National Institute for Medical Research in London, UK, and his postdoctoral training at the Laboratory of Parasitic Diseases, National Institute of Health in Bethesda, USA. He has a long track record as professor of microbiology, pathology, and immunology at very prestigious American universities such as Cornell University, University of Pennsylvania, and Washington University School of Medicine. In 2015, he was recruited as professor at the University of Freiburg and as senior group leader at the Max Planck Institute of Immunobiology and Epigenetics in Germany. Dr. Pearce's laboratory focuses on the role of cellular metabolism in immune cell function and fate during infection and cancer, in order to identify ways to inhibit or promote metabolic pathways to manipulate immune responses. His work has been published in the most prestigious journals such as Nature Immunology, Blood, and Journal of Experimental Medicine, among many others. And given his excellence in research and teaching, Dr. Pearce has recently joined Johns Hopkins University in Baltimore as a Bloomberg Distinguished Professor from the Max Planck Institute of Immunobiology and Epigenetics. Okay, thank you very much, Dr. Pearce, for joining us today. The title of his presentation is Cytokine-induced expressions of amino acids transporters in immune cell activation. Well, Dr. Pearce, again, thank you for accepting our invitation, and the floor is yours.
Okay, well, thanks so much for the invitation to speak today. It's a pleasure and an honor. And I noticed as I was about to come on the camera, I was checking back over the emails that I received, and early on, one of them mentioned that you would like me to speak about macrophages, but I'm afraid I'm not going to do that today. I'm going to talk about activation of plasmacytoid dendritic cells. I hope that's okay, but I suspect the general area that I'm talking about will be relevant in macrophages as well, although there may be subtle differences. As you're all probably very much aware, plasmacytoid dendritic cells are known for their ability to produce type 1 interferons. And these cells are found in the blood, they enter tissues, they respond through endosomal TLRs 7 and 9 to nucleic acids by switching on the expression of genes encoding type 1 interferons. And they're supposed to be one of the major producers of these cytokines in the body. It's also apparent that these cells also make a lot of TNF and chemokines, although that's sort of not discussed as broadly. And while these, I'm just setting my clock starting here. While these cells are clearly must have important roles in immunity to infection and to other insults, they nevertheless are sort of very well known for their association with autoimmune diseases, autoimmunity and autoinflammation, where type 1 interferon signatures are so prominent. And in particular, there's been a lot of interest in the role of PDCs in systemic lupus, where type 1 interferons appear to be very important. So it's from that perspective of really trying to understand how plasmacytoid DCs become activated that we've become interested in these cells because we thought it would be, the more we can understand about how they get activated, the greater the chances that we'll be able to interfere in those activation steps and therefore, disable these cells in settings where they're associated with disease. And obviously, we've been looking at this from a metabolic standpoint because that's the sort of focus of the lab. So what we ended up focusing on was the question of the biology of interleukin-3 for these cells. And I should say that, in the past, we began, our interest in these cells sort of began because we worked at WashU and our labs were close to Marco Colonna's and Marina Cella's. And they, played such an influential role in discovering PDCs. We heard a lot about them and we became interested in the metabolism, but we were working on mouse cells primarily. And there were certain sort of difficulties with working with mouse cells. It's hard to get enough of these cells because they're quite rare. And the cells are not terribly robust when you take them out of the mouse. When we moved to the Max Planck Institute, a new postdoc, Katerina Jez, who did the work that I'm going to talk to you about today, joined the lab from Doreen Cantrell's lab and was interested in studying these cells, but wanted to work on human PDCs because, well, on the one hand, they're easier to get because you can just get blood from the blood bank and isolate the cells from the blood. They're still pretty rare, but you can get them. And secondly, of course, you're that much closer to working in a relevant system because it's human autoimmune diseases that we're really interested in. So we started to work on human PDCs, and one of the really interesting things about human PDCs that actually sets them apart to some extent from most PDCs is their dependence on interleukin-3. So human PDCs express the interleukin-3 receptor at high levels. And, from the earliest days, people realized that to keep these cells fully functional in vitro, you had to add interleukin-3 to the cultures. So, routinely now, when people work on human plasmacytoid dendritic cells, they grow them in culture with human IL-3. They culture them with IL-3. There are also strong links between elevated levels of IL-3 and incidence of lupus. So there's a link from patients between IL-3 and SLE. And consistent with this, there's also a link between the IL-3 stimulated gene signature and the type 1 interferon signature as well, suggesting that there's some relationship between the two. Although the receptors certainly are distinct and the signaling pathways are distinct. But nevertheless, there's this overlap between the two in terms of the genes that are switched on in cells by exposure to these two groups of cytokines. And what struck us particularly interesting about this was that IL-3 is really a prototypical cytokine that's known to act as a growth factor to promote anabolic metabolism. And these observations were made early on, about 20 years ago, in Craig Thompson's lab when he was at the University of Pennsylvania by Matt van der Heijden, who was in the lab at that point. And they were working on a cell line that they made IL-3 dependent, and they really worked out what IL-3 was doing in those cells. And one of the things it was doing was switching on anabolic metabolism that was necessary for the cells to continue to proliferate in their work. So all of these things put together drew our attention to this cytokine and made us interested in it. Okay, so to begin working on this, we obviously wanted to establish a system first in which we were able to get the cells out of human blood and see that we could activate them in vitro and have a good system for measuring their activation. And so what we did here was we took we sorted PDCs out of peripheral blood using the Miltenyi Max kit for isolating these cells, and then we cultured them in vitro either with IL-3 alone or with the addition of CpGA for various amounts of time, and then measured TNF and interferon alpha production using intracellular staining for these cytokines. And on the left, you can see the sort of flow plots, and on the right, a bar graph showing cumulative results from multiple experiments. And what you can see is that the cells actually begin to make, before they make type 1 interferons, they start to make they start to make TNF. And then they start to make type 1 interferons. And by about 12 to 18 hours after you stimulate the cells, basically the cells have start to switch down. And by 24 hours after you activate them, everything's finished. They've made their cytokines. And you can see cells that, make both cytokines. Some cells only make type 1 interferon. But it seems that every cell that makes type 1 interferon also makes TNF in this kind of experiment, at least at the beginning. But the point is, we had a system here for studying the activation of these cells using a system which allowed us to target individual cells using flow cytometry. And this is simply to show that as these cells become activated to make interferon alpha and TNF, they also start to express genes encoding a lot of different chemokines. And in this experiment, we're measuring expression using this prime flow technique, where you basically use a labeled oligo to detect messenger RNAs in a specific fashion inside cells, and then use a second label, a fluorochrome coupled label, to be able to identify binding of the chemokine messenger RNA specific probe. And so you can identify cells that are actually expressing these genes. And you can see that there's significant expression of at least this subset of chemokines here as you activate the cells with CpGA in the presence of IL-3. So these cells, actually make a lot of different mediators that can affect the milieu that they find themselves in. So to examine the role of IL-3 in this, we set up a system which was very simple. We took peripheral blood mononuclear cells, sorted PDCs, rested them overnight in the absence of IL-3, and then stimulated them with IL-3 in the presence or absence of CpG, and then looked at their activation, absent IL-3 or with IL-3. And what we saw was that over the course of 5 hours, there was no difference in the viability of cells that were stimulated with CpG in the absence or presence of IL-3. But if we were to look out beyond that, then we started to see that the cells that were stimulated in the absence of IL-3 would begin to die more rapidly than the cells that were stimulated with IL-3. So certainly, as you take the cells out further in culture, IL-3 is an important survival factor that they need in order to continue to live in culture. But most of our assays that I'll show you, in fact, nearly all of them, were done using intracellular staining for various factors at 5 hours when viability was equivalent with or without IL-3. And anyway, in all of our experiments, we gated on living cells. So the results I'm going to show you are not a reflection of anything to do with increased mortality due to the absence of IL-3. So this was the sort of initial finding, and that was that if you look at TNF and type 1 interferon production and stimulate cells that are not cultured in IL-3 with CpGA, they make very little of either cytokine. But when the cells are cultured with IL-3, then they respond strongly to CpGA by making both TNF and type 1 interferon. And that's illustrated on the right here in these bar graphs. And what these results suggest is that IL-3 is necessary to prime the plasmacytoid dendritic cells to be able to respond to stimulation through a TLR, in this case by CpG, which is a TLR9 agonist. So IL-3 is important for priming these cells to be responsive to TLR stimulation. And that's sort of illustrated here, now looking at not only at the protein, but also at the messenger RNA that encodes the protein, using a combination of cytokine intracellular staining and messenger RNA encoding the cytokine staining using prime flow. And what you can see is that without IL-3, CpGA stimulates very little interferon gamma expression, either at the messenger RNA level or the protein level, and that you need IL-3 in the cultures for CpGA to be able to induce transcription and translation of type 1 interferon. The situation is slightly different with TNF, because it turns out that IL-3 alone is able to stimulate the transcription of the TNF gene. And this is something that we weren't expecting, but the result is quite clear compared to here, where there's a few of the cells express the TNF messenger RNA. If you add IL-3, you can see that nearly all of them start to express TNF messenger RNA. But to get this translated, you need to add CpG. So the addition of CpG allows then the TNF messenger RNA to be translated into TNF protein. And CpG alone, in the absence of IL-3, really doesn't do very much at all. So IL-3 is important then for the transcription and translation of type 1 interferon, and in response to CpG. And the same is true for TNF. But there is this subtle difference between the two in that TNF transcription is induced by IL-3 alone, although the production of the protein requires IL-3 and CpG. So, as I mentioned earlier, IL-3 is sort of really the prototypical cytokine which acts as a growth factor to promote anabolic metabolism based on the work of Craig Thompson and his colleagues. And we know from the work of Sabatini and others, and I sort of draw your attention to this review by Brendan Manning here, that anabolic metabolism is regulated centrally by mTORC1. And we knew from work from Bali Pulendran's lab that mTORC1 is important for PDC activation because rapamycin, a selective inhibitor of mTORC1, was able to block type 1 interferon production by PDCs stimulated with CpG in work that Pulendran published quite a few years ago now. So we asked whether or not, in fact, IL-3 was working in PDCs to promote mTORC1 activity in order to allow them then to be able to respond to CpG and produce cytokine. So just a very brief reminder, it's a bit fuzzy this, I'm sorry. I borrowed this figure from this review, that mTORC1 is a central regulator of metabolism. It's activated by growth factor receptors, insulin receptor, through a variety of different mechanisms. Its activity is dependent on there being sufficient amino acids in the cell. This is important, I'll get back to this shortly. And when activated, mTORC1 both promotes translation of messenger RNAs through its ability to regulate S6K and 4-EBP1. And it also drives a metabolic pathway that's associated with anabolism, including enhanced glucose metabolism, which then feeds into lipid synthesis and nucleotide synthesis. So really it is at the sort of center of the sort of areas that we think are engaged in activated PDCs. So you can measure mTORC activity by using, again, intracellular staining for phosphorylated S6, which is phosphorylated by S6K, which is one of these targets of active mTORC1. And what we found was that in PDCs stimulated with CpG plus IL-3, a significant percentage of the cells have active mTORC1 as measurable by phosphorylated S6, and that's shown here. This is largely dependent on CpGA, because if you just stimulate the cells with IL-3, you don't see this. And if you stimulate the cells with CpGA without IL-3, you see a little bit of phospho-S6, approximately a doubling compared to baseline, but nowhere near as much as you see in cells stimulated with both CpGA and IL-3. And this phosphorylation of S6 is mTOR dependent, because if you, on the bottom here, if you feed in rapamycin, the specific inhibitor of mTOR, you can see that you can significantly inhibit the number of cells, the percentage of cells which have phosphorylated S6 in them. So this CpGA and IL-3 driven activation of mTORC1 is occurring in these plasmacytoid dendritic cells. And as shown by Pulendran's lab, this is important for cytokine production, because if you add into these cultures rapamycin, you see that you're able to significantly inhibit the production of interferon alpha and TNF. So this is all consistent with what has been published previously. Now, what we, because of its link to metabolism and messenger RNA translation, we examined these PDCs that were activated with IL-3 and CpG to see if we could measure changes in those two parameters. This is an assay that measures the incorporation of puromycin as an indicator of translation. And what we saw there was when we took PDCs and activated them with IL-3 plus CpGA, we saw an increase in translation, which would be consistent now with the ability of these cells to, the fact that these cells are translating lots of messenger RNAs that are being produced as a result of activation and making proteins that are destined for export. And they're making those in quantity. And that's what we think we're measuring here. But if you feed in rapamycin or taurin, which is another inhibitor of mTOR, you see that translation is significantly inhibited. And it's inhibited almost as much as if you feed in cyclohexamide, which is a specific inhibitor of translation. So yes, we do see increased translation in these activated PDCs, and this is all dependent on mTORC1. And we also see increased metabolism in these cells. And what we're doing here is we're measuring extracellular acidification rate. And this is a mark of the production of lactic acid by cells, which is an indicator of engagement of Warburg metabolism. And what we found was that very shortly after adding CpGA to PDCs cultured in IL-3, we saw an increase in extracellular acidification rates. And this was sustained over the time course that cells are producing cytokine. So the cells change metabolically when they become activated. And this type of Warburg metabolism is strongly correlated with anabolism. And so this is consistent with the role of mTORC and what's happening there. And this increase in glycolysis is also important for activation of the cells, because if we inhibit the central enzyme in glycolysis, GAPDH, with a specific inhibitor of GAPDH, heptalytic acid or koningic acid, we can see that we can inhibit cytokine production by these PDCs that were stimulated with CpG plus IL-3. So then, just to conclude, we think that when we activate PDCs, the cells require IL-3 to be primed, to be able to respond to CpG, to become activated. And this process requires mTORC1. And mTORC1 is heavily regulated in a metabolic change that occurs in these cells that's required for activation, this increased glycolysis, and also heavily involved in the ability of these cells to start translating all these messenger RNAs that they're making as a result of the activation stimulus. So I'd like now just to take you back to that diagram I showed you from the review by Sabatini, and just now turn to this requirement for certain amino acids to allow mTORC1 activation, and in particular leucine. And this is because there's a sensor of there are sensors of amino acid concentration in the cell, and these essentially will only allow mTORC to become activated when there are sufficient amino acids around for the cell to be anabolic and start translating lots of messenger RNAs to make protein, because you can't do that unless there's sufficient amino acids available. Now we know that leucine is acquired by cells through something called a system L amino acid transporter, or LAT. And this system L amino acid transporter, SLLA, is in fact a heterodimer of two proteins, SLC7A5 and SLC3A2. SLC3A2 is also CD98. And you need these two proteins in order to take up leucine. And work in Doreen Cantrell's lab showed that you could measure SLAA activity using kynurenine. And kynurenine is autofluorescent, and it's taken into cells through this SLAA. And that's just fortuitous because it allows you experimentally to add kynurenine to cells and then measure any increase in fluorescence, and using specific inhibitors to make sure that you're looking at this amino acid transporter, you can take this approach to get a good measure actually of SLAA activity. And so we hypothesized that what IL-3 might be doing, in fact, was regulating the expression of either SLC7A5 or SLC3A2. In other words, regulating the expression of this amino acid transporter, and in that way, acting to regulate mTORC1. And that was, in fact, we hypothesized that this was the priming event that IL-3 is responsible for allowing these cells to then become responsive to CpG. So we examined that using, again, this prime flow technique where you can measure messenger RNAs that are coming from your genes of interest. And we look for messenger RNAs encoding SLC3A2 and 7A5, the two components of this SLAA. And what we found was that PDCs at rest, about 30% of the cells express both of these chains. The expression that seems to be linked, and that you don't really see cells making one without the other for the most part. That's not completely true, but it's mostly true. And if you stimulate these cells with CpG, you see some increase in this population. You can see that from here to here. But the major stimulus for the expression of these two components of the amino acid transporter is, in fact, IL-3, because basically all of the cells express both of these genes when you stimulate them with IL-3 alone. And when you add CpGA, you don't see any increase. And so the drive to express this transporter is coming from IL-3. So IL-3 is inducing the expression of this amino acid transporter that imports amino acids that are required for mTORC activation. And that's illustrated here, taking the means from various experiments. And you can see that cells grown without IL-3 or CpGA, very few, low percentages express the two chains of the transporter. You add CpGA alone, you see a slight increase. But when you add IL-3, nearly all of the cells start to make this transporter. And is the transporter active? Well, we were able to measure that using this kynurenine uptake assay that I mentioned to you. And what you can see here quite clearly is that in cells stimulated with IL-3, basically all the cells are able to take up kynurenine compared to only a small percentage of unstimulated cells. And that adding CpGA on top of the IL-3 doesn't increase that any further. So IL-3 stimulates the expression of a functional SLAA that can take up kynurenine and therefore leucine. And this is specific because BCH is an inhibitor of the SLAA, and that completely blocks kynurenine uptake in these experiments. Okay. So what does kynurenine uptake or leucine uptake got to do with the rest of the events that I've been showing you? Our hypothesis was that it would be the cells that could take up leucine that were basically primed to be responsive to CpG. And we believe that is the case, and that's illustrated here in this experiment where we're looking at phosphorylation of S6, which you remember is this mark of activated mTORC, in the context of kynurenine uptake. And here you can see in untreated cells, there are some cells which express phosphorylated S6 and some that take up kynurenine. If you add CpGA without IL-3, you don't see much of a change in these percentages. But when you add IL-3, you now see that basically all the cells start to take up kynurenine, but none of them are strongly positive for phosphorylated S6. But now when you add CpG on top of that, you see that a large percentage of the cells now are kynurenine positive and positive for phosphorylated S6. So these are the genuinely, metabolically activated plasmacytoid DCs. And these are, in fact, the cells that are making cytokine, if you backgate on these. And I'll show you examples of that as we go forward. And this event of being able to take up leucine is really critical for type 1 interferon production, because if you add a specific inhibitor of LAT, it's a small molecular inhibitor called JPH. There are a few numbers after that. I can't remember what they are right now. JPH something or another. That really markedly inhibits type 1 interferon production. And it's a sort of dose-dependent effect. And you can see a similar thing if you deprive the cells of leucine. So take leucine out of the culture, and now the cells are much less capable of making type 1 interferon in response to CpGA, even when they're IL-3 primed. Okay. So quite a lot is known about IL-3 signaling pathway. And, we know that IL-3 signals through the IL-3 receptor alpha and something called the common beta chain to activate JAK2 and then STAT5, and phosphorylated STAT5 drives the expression of many of these genes that are part of the IL-3 induced signature. This common beta chain here is shared with the GM-CSF receptor and the IL-5 receptor, although those two receptors have different alpha chains, which allow them to respond only to those two cytokines. But nevertheless, this is the receptor for IL-3. And if you look at if you look at the and we did this using available data that have been published, it's clear that both SLC3A2 and SLC7A5, the two components of LAT, SLAA, that are induced by IL-3, both of these have STAT5 binding sites in their within their upstream sequences or within the body of the sequence, the codon sequence. So we there's evidence from these binding sites that STAT5 would drive the expression of these two genes, which is consistent with what we were seeing. And furthermore, what we found was that when you activate the cells, you see that with IL-3, you see that STAT5 becomes phosphorylated, consistent with this. And that's shown here. In untreated cells, there's a small percentage of the cells contain phosphorylated STAT5. CpGA doesn't really change that. But now, basically all of the cells stimulated with IL-3 have phosphorylated STAT5 in them. That's consistent with this pathway. And you can inhibit that phosphorylation using these two drugs here, AZD, which is a JAK2 specific inhibitor, and baricitinib, which is a JAK1/2 inhibitor, a drug that can inhibit both JAK1 and JAK2, whereas this is a JAK2 specific inhibitor. And what you can see is that both of these basically inhibit the expression, inhibit the phosphorylation of STAT5 driven by IL-3. Now, we're interested in these two inhibitors because this one in particular, baricitinib, is in clinical use and was shown a couple of years ago in a clinical trial to be effective in limiting against certain aspects of lupus. So there's promise for this drug, which inhibits JAK1 and JAK2 in the clinic. And this drug, which is not available clinically, which is the JAK2 specific inhibitor, really, in terms of what we're looking at, mimics everything that baricitinib does. But these are drugs of relevance from the perspective of their potential use to treat autoimmunities, which are associated with plasmacytoid dendritic cells. So obviously, we're interested in whether or not these effects here are linked to the inhibition of function of the PDCs. So these effects of the important effect of IL-3 in these cells, of course, is to drive the expression of the two genes which encode the two components of the SLAA. And when we add either AZD or baricitinib, we see very significant inhibition of the expression of both of these genes. And that's consistent with the fact that AZD and baricitinib are inhibiting the signaling pathway that's driven by IL-3 by blocking JAK2. And this is just a sort of graph which sort of shows the results of multiple experiments of this type. And if you look down here, you can see that when you add these two drugs to these cultures, they in fact inhibit phosphorylation of S6. And of course, we've argued that the expression of this amino acid transporter is essential for mTORC1 activation, which we're measuring as S6 phosphorylation. And that's occurring in cells which are stimulated with IL-3 and CpG, and it's linked, if you remember, to cytokine production. But when we add these inhibitors of JAK2, we see inhibition of S6 phosphorylation, suggesting that these two drugs here, which target JAKs, are consequently inhibiting mTORC as well in the system. And if we look at more detail of this, what you can see is that when you add CpG plus IL-3, cells start to take up kynurenine, and it's those kynurenine positive cells that express the cytokines, as I mentioned earlier, interferon alpha and TNF. But when you add azacitidine, you see a shift to the left. A shift to the left of this population here, because a lot of these cells are no longer taking up kynurenine. And this gate here, which previously held the cytokine positive and kynurenine positive cells, is now negative, because these two drugs are potent inhibitors of activation in PDCs because of their effects on the IL-3 signaling pathway. So I'm going to change tack a little bit here now, although I'm going to keep a connection in what I'm going to tell you with what I've already told you. But I'd like to just tell you about some results that we have that sort of tie what we're seeing in vitro to what's happening in patients with lupus nephritis. And these results come from experiments in which originated from this observation here. And I think this is the first piece of data that I showed you at the beginning of the talk, and that is when you stimulate purified PDCs with CpG and IL-3, you see at least 4 distinct populations of cells. You see cells that are making TNF plus interferon, TNF alone, or neither cytokine, or a set of cells that so that's it. So those groups there. And so we wondered whether this heterogeneity in the PDCs was restricted to this kind of thing here, or whether in fact it was a broader heterogeneity. And of course, one way to look at that was to do single cell RNA-seq. And so what we did was we sorted PDCs based on the expression of these two markers here, one of which is the IL-3 receptor, which is a very strong marker of human PDCs, as I've mentioned before. So before purification, a very small fraction of peripheral blood mononuclear cells are PDCs, but after purification, we've got a very pure population. And we sequenced these cells, and we found that there were 12 distinct clusters of cells that were transcriptionally distinct from each other. And that one of these clusters, cluster 9, actually was only present in cells that were stimulated with CpGA. So this is kind of interesting to us, and I'm not going to go into it too much any further here, but it is interesting that these cells that we previously would have considered to be homogeneous are in fact heterogeneous. I don't know exactly what this means, although we're working on that. But nevertheless, when we activate these cells with CpG, we see the emergence of a new cluster of cells closely related to cluster 2. And in fact, that's illustrated over here, where we're looking at the percentage changes in these clusters. And you can see that, fraction 9, basically that appears as a result of CpGA stimulation. Some of these other clusters go up and down. But when we look in these different clusters for type 1 interferon production, you can see that cluster 9 is really the type 1 interferon producing cluster. This is cluster 9 here, and we're looking at expression levels of 3 different type 1 interferons, alpha, beta, and lambda. So it's really cluster 9 that's making these cytokines. And also, to some extent, cluster 2. And cluster 2, it turns out, shares quite a lot of similarities with cluster 9, and it's sort of tempting to think that cluster 9 emerges from cluster 2 as the cells become activated. And you can see that cluster 2 is actually increasing a bit as well as a result of activation. So that's the situation. And then what we did was we compared these single cell transcriptomes from our in vitro activated PDCs with a single cell RNA sequence data set, which was made from cells isolated from lupus nephritis kidney biopsies, right? And this is a paper by Arazai et al. That was published a couple of years ago, and we refer to these as SLE PDCs. This is a great paper in which they basically pulled out all the immune cells from these diseased kidneys, and they single cell sequed them all. And one population in there, population 22, shown here, was the PDCs. And there were only a few, there weren't many of these cells in the lesion, but they were there, and they got sequence on them. And when we looked at in all of the different cell types, and there's the gamut of cells here, B cells, T cells, various other immune cell types in these kidneys. When we looked at the expression of IL-3 receptor alpha, the common beta chain, and the two components of SLAA, SLC7A5 and SLC3A2, we found that, in fact, it was the PDCs which were sort of really strongly expressing these 5 genes or 4 genes that we knew to be important for the IL-3 different induced effects in PDCs that are important for activation. So these are cells now that are coming out of a patient that has disease, and so this was quite exciting to us. And so then what we did was compare gene expression in these SLE PDCs with genes which are differentially expressed, and we're talking about differentially expressed genes now, in cluster 9 and cluster 2, which are these 2 clusters from our in vitro seeked PDCs that contain the type 1 interferon transcripts. And we looked at these 2, and we looked within these clusters for differentially expressed genes that were shared with the SLE PDCs. So we were looking now for a signature in these SLE PDCs that we could relate back to what we were seeing in vitro, to try and identify new genes that were associated with this activation pathway that we'd defined. And we found that amongst the 3 different populations, cluster 2, cluster 9, and these SLE PDCs, there really were only 3 genes that were differentially expressed in vitro that fitted with the SLE PDCs, and these were these 3 genes here. And you can see they were expressed in cluster 9, and to a greater or lesser extent in cluster 2 as well. And there was some expression in other clusters as well, but you can see that the prominent signature is in cluster 9, which are the activated type 1 interferon producing PDCs. So now I'm going to focus just for a couple of slides now on this gene here, ENP22, which turns out to encode a gene called, to encode a protein called autotaxing. And many of you will be familiar with autotaxin. This is an enzyme that catalyzes the formation of extracellular lysophosphatidic acid. This is a lipid mediator, which is known, which works through a number of different G protein coupled receptors that are arranged, at least 6 GPCRs that can respond to LPA. And LPA has been reported to have multiple sort of functions, but it's able to evoke growth factor-like responses in cells. And these effects are thought to be important in a variety of diseases, including autoimmunities, like arthritis and systemic sclerosis, and in colitis, and in cancer progression. So that's my 40-minute timer, but I've only got a couple of slides left, so I'll finish shortly. And so what we found was that, so we looked at ENPP2 expression in a little bit more detail. And what we found was that ENP22 expression is dependent on, and I'm just noticing there's a mistake here. This should not be here. this should be absent, that cross there, because you need both IL-3 and CpGA to induce expression of EMP22 in vitro. And both AZD and baricitinib, which are those JAK2 inhibitors, are able to inhibit expression of EMP22 in these PDCs that are stimulated with IL-3 plus CpG. And also, expression of EMP22 is dependent on mTOR as well, because taurine is able to inhibit expression of EMP22. So much of the pathway that I've explained to you before, that's in which the IL-3 primes the cells to be responsive to CpG, and in which mTOR is implicated, is important for the expression of this gene here in vitro, which is shared with these PDCs, which were pulled out of a disease site in a patient. And what we can see is that in these cells, EMP22 expression is really associated with the expression of type 1 interferon. And this is a slightly different type of plot in which we're looking at EMP22 and interferon alpha expression, looking at messenger RNAs using prime flow in cells which were stimulated in these different ways in the presence or absence of AZD or baricitinib. And the different colors reflect EMP22 negative interferon positive cells, EMP22 EMPP2 positive cells, interferon alpha negative cells, and cells which are double positive, those are the green cells. And you can see that these green cells only appear when you add CpG plus IL-3, and you can inhibit their appearance with these inhibitors of JAK2. And and it turns out that there is a small molecule inhibitor of autotaxin called ziritaxestat, or ZIA for short, and that's able to inhibit cytokine production and activation by CpGA in PDCs. And I'll show you the data to support that here. Here we're looking at cells now which were stimulated in these various ways with or without this inhibitor. And we're looking at phosphorylation of S6 and kynurenine uptake. Here, when you stimulate with IL-3 alone, you see S6 phosphorylation. You add CpGA, and now you start to see a population of cells that's S6 positive and taking up kynurenine because they're expressing the amino acid transporter. And when you feed in this inhibitor of autotaxin, you see diminished percentage of cells that are double positive. And then when you look in that population there, the green population, which is the top right-hand corner, it turns out that that's really the only quadrant in which you have cells that are making type 1 interferon, that's shown here, or TNF, and that when you add this inhibitor of autotaxin, you see a dose-dependent inhibition of production of both of these cytokines. So in summary then, this is what we think is happening in these cells. We know that they are dependent on IL-3 to become activated, and what IL-3 is doing is it's working through its canonical signaling pathway which leads to the phosphorylation of STAT5, which then induces the expression of both genes encoding the system L amino acid transporter, SLC7A5 and SLC3A2. And this then makes PDCs, basically all the cells express this transporter. And these cells then are primed to be able to respond to TLR9. Without this step, they can't respond to activation through TLRs, but once they've been primed in this way, they can. And a subset of these cells then becomes activated. Not all of them, for reasons that we don't know. And we appreciate that this population is heterogeneous, and a subset, a subpopulation of cells becomes activated. In these cells, mTORC1 is active. You see metabolic activation, and the cells begin to express genes, including EMPP2, which encodes autotaxin, and make these cytokines, which are implicated in autoimmune disease development. And what we found is that you can really target the production of these cytokines at multiple different levels by targeting components of this sequencing pathway, signaling pathway. And that includes using inhibitors of JAK2, inhibitors of the system LAA amino acid transporter, inhibitors of mTORC1 or the glycolysis pathway that is upregulated as a result of the activation of this central regulator of metabolism, or in fact inhibitors of ENPP2 to block the production of cytokines. Now, while we understand really how these three pathways are working, we think it's really not clear to us yet about how ENPP2 is working beyond the assumption that it's working through its ability to make lysophosphatidic acid, but that requires a lot more work for us to really get to the bottom of that. Okay, so I'd just like to finish by acknowledging the people that did this work and the funding that we received that supports this research. So most of this work was done while the lab was at the Max Planck Institute in Freiburg, and it was done by Cassia Jez with David Sanin for the most part, although it was in collaboration with all these other people that are on this slide. And Cassia and David are in the process of moving now to join me at Johns Hopkins in Baltimore. And this is an aerial view of Baltimore from a bar on the top of a hotel by the harbor, where we were having a couple of drinks a couple of days ago. It's very pleasant. And then these are the other people at the Max Planck who were part of the team there, who won't be joining us in Baltimore, but Aga Kabat was also part of this project, and Michael Stanzak, and they are either here, in Michael's case, he works in Erica's lab, or in the process of moving here to continue the work. And we had, the benefit of some great collaborators on this project and funding from these societies and agencies here. So thanks very much for your attention, and I'm sorry I went a little bit long, but I'd be pleased to take questions if you have any. Thank you.
Okay, thank you very much, Dr. Pearce, for your excellent talk. You have really made this complex scenario very simple to understand, so thank you. So yes, we have here some questions. Let me check. So first, one's a really simple one, but which are the in vivo source of IL-3, maybe in the context of autoimmunity and also in the context of viral infections?
Yeah, so that's a great question. So, IL-3 can be made by CD4 T cells, but it can also be made by stromal cells like epithelial cells. And what I didn't show you, but which we know, is that actually there's IL-3 in the bloodstream, even of healthy patients. And that to some extent, the IL-3 signaling pathway is already activated in PDCs in the blood. When you take them out and you rest them overnight in vitro, that signaling pathway switches off, and you can reactivate it in vitro by adding IL-3 to see the kinds of things that I showed you. But in vivo, some of those IL-3 dependent events are already apparent in circulating PDCs. Now, a slight complexity is that both GM-CSF and IL-5 can also signal through this common beta chain. And we know that GM-CSF really has overlapping functions with IL-3 in everything I've shown you today. The PDCs don't express the IL-5 receptor. So it could be that GM-CSF is doing it. And then we think that when cells move into a tissue, they need to see IL-3 to continue to be responsive. And we don't know where that's coming from. It could be coming from T cells or stromal cells, or it could be, in fact, GM-CSF that they're seeing in order to be functional. We just don't know at this point, but that's the situation.
Okay, so next question is whether have you seen if ENPP2 is also related to interferon lambda, and in relation to that, it is mentioned that interferon lambda is associated with mucous immunity. So do you think that these pathways could be operating to particular 9, IL-3, ENPP2, interferon lambda in the mucus that could be more microbial stimuli?
Yeah, presumably this pathway is important for the beneficial effects of PDCs, right, in immunity to viruses or in other settings. So I think it is. It's a fairly recent discovery, this one about the ENPP2, and we really haven't done any targeted experiments to explore its function, how it's working. I'm interested in that. But I assume that all this is important for the activation of the cells in settings where they're doing positive things or they're doing negative things. So, it just so happens that the slant I've taken in the talk is to talk about, their involvement in autoimmunity and trying to block the pathway. But you could equally as well talk about trying to promote this pathway if you were thinking about immunity to a virus or some other situation in which these cells are important.
Okay, here is another question. Let me see. Sorry. Here it says that IL-3 and CpG treated cells express AAC transporters around 85%, but still fully activated PDCs are around 50%. So could it be explained in terms of heterogeneity, of PDCs?
Yes, it could. So, I mean, this is something we haven't fully got to grips with yet, but yeah, it's clear that IL-3, all the cells get activated basically to express the amino acid transporters, but only a subset of them can then respond to TLRs and get activated. And it's the same if you use other TLR agonists. We've looked at, TLR7 and other types of TLR9 and agonists as well. And it's always the same. It's only a subset of the cells that really start to make cytokine. So yeah, there is heterogeneity in there. And I don't know, we have to try and understand that in more detail as to why it is that all the cells don't make cytokine. I don't know what the answer to that is.
Okay, so next question is the activity of IL-3 promoting the transcription of this cytokine is specific, or is it also observed with other cytokines?
Well, GM-CSF is the other cytokine that basically does exactly the same thing, as far as we can tell, as IL-3 in terms of what I was describing today. It may be the GM-CSF does other stuff as well, but in terms of inducing the expression of the amino acid transporter, it's the same as IL-3. It's overlapping, and it's all dependent on STAT5 and all the rest of it. I think it's because they share the common beta chain,
Yes.
Yeah.
And here, perhaps you have already addressed this, but it is a requirement of IL-3 for the survival of PDCs in vitro expect to be needed in vivo. And linked to this, does IL-3 also have this promoting role in a context of viral infections?
Yeah. So I don't know. I feel, we haven't done anything on viral infections at all. And we need to do that. And I think it does have this role in vivo. And the reason that I say that is that we have looked at PDCs from patients who are on a JAK inhibitor, right? People who have rheumatoid arthritis and are on a JAK inhibitor, compared to people, healthy individuals. And in those patients, when you take those cells out, you can see that their effects on the PDCs from the person on the JAK inhibitor that look very similar to what we're seeing in vitro. So I think that's an indicator that's, JAK1, JAK2, STAT5 is important in vivo as well. That's how I take it anyway.
Okay, so we have one further question concerning, since CD98 mediates coupled glutamine efflux and used in uptake. The question is whether have you studied whether there is loss of glutamine in these cells, and if it alters any function in PDC.
Yeah, so that's a good question, and we haven't really carefully explored the function of glutamate, and I'm sure it's important because you know, I didn't really show it in any detail, but you know, inhibition of glucose metabolism doesn't really block activation fully. I don't know if you, I mean, maybe you picked that up. I showed a figure of what happens when you inhibit GAPDH, and there is significant inhibition of type 1 interferon production, but it doesn't shut it off. And the cells remain capable of producing more cytokine than resting cells, you know, so they're still activated and they're still making cytokine. And what that applies to me is that, you know, there are other metabolic pathways there that can support cytokine production. And I think glutamine is important for those. And probably, you know, there are other things going on in the cells that are really making glutamine important. And one of the things is, well, I won't go there. I won't go into it because it gets a bit complicated. But yeah, I think glutamine is important, and we're trying to do some experiments on that at the moment to get to the bottom of it.
Okay, so thank you, Edward. Please, I think we are on time.
I would like to add.
Okay, sorry.
No, just I want to know whether you suspect or have you addresses whether this lipid mediator LPA, which is produced by these enzymes that you related with. Do you think that this lipid mediator is at some point related with the activation of cytokine production, or in, I don't know, in an autocrine or paracrine way?
Yeah, so I don't know what's going on there. You know, first of all, I don't know whether, you know, this inhibitor, ziritaxestat or whatever it is. I don't really know. We haven't done any molecular targeting of the enzyme, so we, you know, and we haven't used any other inhibitors. That's sort of a, you know, an initial look-see. So I think we have to, we're cautious of that result a little bit. And I think that one has to imagine, from what I can understand so far about autotaxin activity, is that it makes lysophosphatidic acid in the extracellular environment, and then the effects on a cell of lysophosphatidic acid are through GPCRs. So one imagines then that some GPCR, and there are many of them for lysophosphatidic acid, is signaling in some way to contribute to the activation pathway that drives type 1 interferon and TNF production. But I don't know what that pathway is. And that's something that, you know, I just don't know what it is. So we have to look into that. And we have to, you know, we really have to further establish the basic observation as well, and work on that a little bit more.
Thank you.
So one more question, if that's okay with you.
Right, yeah.
Have you studied RAG and REV proteins in PDCs stimulated by IL-3?
No, we haven't.
Okay, so we have a lot of congratulations in the chat, and thank you so much for your talk.
Thank you.
Mariana.
Yes,
thanks again for inviting me. Thank you.
I have light here. Okay, well, thank you so much, Dr. Pearce, for your excellent talk. Really, we appreciate your time and your effort for being here, and we hope that someday we can have you in person here in Argentina. So, thank you so much. And para todos los asistentes, muchísimas gracias por habernos acompañado. Se terminan estos jueves de inmunometabolismo. Espero que nos acompañen en las próximas actividades de SAI, y muchas gracias a todos por participar. Gracias, Mercedes. Gracias, Luciana. Hasta la próxima.